Bom, pessoal, boa tarde a todos. É um prazer estar aqui novamente para a nossa quarta live dessa série de webinars que a gente está tendo da Reunião Brasileira de Citogenética e Citogenômica. É um prazer ter todos vocês aqui novamente. E hoje eu sou o condutor dessa live, né, dessa discussão, e sou o professor Diogo Cavalcante Cabral de Mello, aqui da Unesp de Rio Claro, e tenho o prazer hoje de compartilhar essa coordenação, esse delineamento dos trabalhos ao longo dessa tarde, né, desse princípio de tarde, com os nossos colegas, o professor Ricardo Gunski, da Unipampa, no Rio Grande do Sul, e o nosso colega Juan Martim Ferro, da Unicamp, aqui no estado de São Paulo, tá? Então, é um prazer, novamente, a gente estar aqui para discutir, né, estreitar um pouco essa distância que a gente tem, pelo menos física, durante esse tempo de pandemia. A gente tem tido sempre seminários muito produtivos. Esse é o nosso quarto seminário. A gente tem tido sempre tardes muito agradáveis e muito proveitosas para discutir um pouco de citogenética, biologia cromossômica no geral e genômica, e hoje certamente não vai ser diferente, tá? Hoje nós teremos a presença da professora Maria Inés Pigozzi, que é da Universidade de Buenos Aires. A professora Maria Inés Pigozzi, ela é licenciada em genética pela Universidade Nacional de Misiones e tem doutorado em ciências biológicas pela Universidade de Buenos Aires. Atualmente, ela coordena o Laboratório de Estudos Cromossômicos, de Estudos Meióticos, perdão, da Universidade de Buenos Aires, e é vinculada diretamente à Faculdade de Medicina. É pesquisadora do CONICET e desenvolve essencialmente pesquisas no estudo de meiose, entendimento da meiose e do complexo sinaptonêmico, com foco essencialmente na primeira divisão meiótica, né, relacionado ao crossing over, analisando justamente os seus mecanismos de ocorrência e quais são as bases cromossômicas que podem influenciar esses eventos que acontecem basicamente em todos os organismos. Novamente, eu agradeço a presença de todos e le doy la bienvenida, doutora María Inés Pigozzi. Estamos muy contentos. Te agradezco muchísimo en nombre de la organización del evento. Estamos muy contentos de ahora tener en este cuarto seminario una persona hispanohablante. Ya estamos más distribuidos en el mundo. Tuvimos gente de hemisferio norte y ahora tenemos una colega que está muy cerca de nosotros y que está aquí en Argentina. Estamos encantados de tenerte y yo estoy seguro que vamos a tener un principio de tarde muy agradable con tu charla. Y te paso la palabra para que empiece tu presentación. Gracias por venir.
Muchísimas gracias. Bueno, si me dan el ok, que están viendo todo correcto.
Perfecto.
¿Me escuchan bien? Bien. Bueno, I'd like to thank first, I choose to speak in English because if I would speak in Spanish, maybe I would speak too fast. And well, I was telling before that it was better a bad English than a fast Spanish, maybe, for the audience. And today, well, I want to first thank to the organizer, Professor Bruski, and because of the invitation and also for the introduction. And I want to say hi to the colleagues that I know personally there in Brazil. And today I'm going to talk about the recombination rates in birds, look through the methods, cytogenetic methods, more specifically using immunofluorescent detection of the crossovers. The recombination rates, understood as the number of crossover events per megabase per generation, are known to vary within species, between species, within individuals, and even between sexes of the same species. In birds, the cytogenetic studies of the recombination were first through the looking at chiasmata in meiotic spermatocytes, metaphase spermatocytes, but also looking at meiotic events, just as recombination nodules in packaging and Lambert chromosomes that are developed during packaging in female birds. All these studies were centered or focused in chicken oocytes or spermatocytes. So all the perceptions and the information that was obtained was biased towards the characteristics of the recombination in the chicken. These first studies showed that the chicken had higher recombination rates than mammals, essentially mice and humans that were used for comparisons, and that there were small differences in recombination levels between sexes. So females and males had essentially equal recombination rates. Some of these features were related to the genome and karyotypic stability that are known in birds, and for this reason, the features of the crossing over in the chicken were considered to be typical for all birds, because we are used to the information of very little variation of genomic size, and also to the conservation of karyotypes, even within in birds with a long history of divergence. So in this talk, I would like to update what is known about the within genome variations of recombination in birds, and also to show how the cytological analysis of the crossing over can help to understand the relationship between recombination rates at different taxonomic scales in birds. Because most of the times that we talk about cytogenetic studies, we use mitotic chromosomes, or even if we look at meiotic chromosomes, we are used to chromosomes at metaphase 1 or at diagenesis. I would like to make a very brief introduction about the structure of the synaptonyma complex. So I'm sure that many of you are familiar with this schematic representation of the recombination of the synaptonyma complex. The synaptonyma complex is almost universal structure in meiotic organisms, and it's composed by two lateral elements that are joined to central elements by transverse fibers that are here in black. The chromatin is attached to the lateral elements, and it's organized by a cohesing scaffold that runs the same way that lateral elements. This structure is very conserved between organisms, but there are now more realistic representations of the synaptonyma complex, and this image belongs to chapter by Professor Hernandez, a researcher from Mexico that has long investigated the structure of the synaptonyma complex. And the same group, in collaboration with Chris Hook from Sweden, they discovered recently more close to a native structure of the synaptonemal complex. Among other things, they showed that the space between the inner surface of the lateral elements is a little wider than thought before, and also that the transverse fibers have an amorphous or non-symmetrical disposition within the central region. And these findings have some implications for the role of the synaptonemal complex to stabilize the interactions between the factors that are involved in the crossing over. Among vertebrates, the most investigated proteins of the synaptonemal complex are those in mammals, especially in mice. In this regard, some of the latest studies of the architecture of the synaptonemal complex were done by Dr. Benavente and his collaborators in the University of Duisburg. And well, the synaptonemal complex in mammals has 8 proteins. The main protein of the lateral elements is CYP3, which is the protein that usually is localized in immunofluorescence studies to study the synaptonemal complex in what we call synaptonemal complex spreads in a surface. And in most of the proteins are located in a complex central region of the synaptonemal complex, and the transverse filaments are formed by homodimers of the CYP1 protein. The group of Benavente recently showed a more real architecture of the synaptonemal complex using expansion microscopy, and they showed that the lateral elements have a helicoidal path. They are not shown here for clarity, but they follow a helicoidal path, and the transverse fibrils don't have a symmetric disposition like in the cartoon that I showed you before. It was the way the synaptonemal complex was seen before these studies. But CYP1 sometimes interacts with another molecule of CYP1 over the central element, but sometimes there are 2 molecules of CYP1 that do not have counterpart on the other side of the synaptonemal complex. I'm just telling these to kind of update and give a small background of the synaptonemal complex in with the most recent information that we have, because the synaptonemal complex, I'm going to show the synaptonemal complex as a framework for the crossover, and I don't want this to give the addition that this is a non-dynamic structure, a static structure that only supports the DNA and the crossover factors. So going more into the analysis of crossing over, the synaptonemal complex for cytogenetic studies is usually observed in micro spreads or surface spreads like this one, because they are more useful to observe all the chromosome elements for the synaptonemal complexes, in fact, in a single nucleus. In birds with the right staining, the recombination nodules, that are these electron-dense particles here, are visible and they are well stained, but that's not the case in all the birds. These are images of pigeon oocytes from Columba livia, the common pigeon. And this, in these birds, the recombination nodules are particularly clearly preserved. But in other birds, like, for example, chicken, recombination nodules are small, and it's difficult to obtain a good amount of nuclei where the recombination nodules are preserved to count the crossovers. So electron microscopy preparations of the synaptoma complex had the advantage of providing a very good resolution for chromosome counts. In birds, this is especially useful because there are many chromosomes. And for karyotype rearrangements, for example, translocations or to study polyploids, and the recombination nodules may be visible with the right condition of staining using phosphotoxic acid, but they are not always clearly visualized. But it's very time consuming because of the methodology. You lose sometimes many nuclei for the observations because, well, I don't want to enter into details, but the electron microscopy grid sometimes hidden the nuclei partially, and you cannot observe them, observe the whole nuclei to count the crossovers. And also the detection of proteins using monogol is laborious and it's not good for quantitative analysis because you have to analyze them at high magnification and then you lose the whole image of the nucleus, what makes it really difficult to quantify the recombination events. With the new microscopy techniques, super resolution offers precisely a good resolution close to the electron microscopy studies. The synaptoma complex has been one of the intranuclear structures more analyzed with this type of microscopy. These are images of a structural illumination microscopy. They were taken by Roberta Ciurano at the Professor Benavente's lab, and they showed beautifully the immunostaining of the lateral elements in chicken spermatocytes, and also immunostaining with a protein of the central region. And that gives me the opportunity to tell you that the antibodies against mammalian proteins recognize the homologs in birds. But this is sometimes um, some of the antibodies against mice proteins of the synaptoma complex are good to detect the components of the synaptoma complexes in birds. But when you go farther in the phylogenetic distances, it's more difficult to use to the antibodies to cross-react because the specific proteins of the SC are not well preserved at this sequence level in phylogenetically distant species. So the super resolution method has a very good good image, but the problem is that it requires a lot of computer processing and also, well, specific equipment and personnel that is trained to handle all the information that is captured with these instruments. So this is not practical, again, for quantitative studies of the crossing over. Then that leaves us with indirect immunofluorescence in synaptoma complex expressed using epifluorescence microscopy. As I told you before, the synaptoma complexes can be detected either by the proteins specific of the axial elements, lateral elements, or by the cohesin scaffold that follows the same path as the lateral elements. And the recombination nodules has a counterpart that are the MLH1 foci that are observed along the SCs. This protein MLH1 is a component of late nodules along with MLH3. And the commercial antibodies against MLH1 that are normally used for studies in non-polypocytic cancer. So there are many commercial antibodies against MLH1 and produce these very neat sharp images of the MLH1 foci. So the studies of crossing overs in birds have been performed using either MLH1 detection in Pakitan oocytes or spermatocytes, or looking at the Lambert chromosomes during diploiding stage. The problem with Lambert chromosomes is it requires extremely skilled from the operator who does the preparations. They're very difficult to obtain. They break easily. So even though they offer a very high resolution for chiasmata, the spreads are often not complete, and most microchromosomes are lost from spreads. So what I'm going to talk about then, the results, is about the crossover detection using MLH1 on synaptonemal complexes in birds. And as it happens in most cytogenetic techniques, the MLH1 focus counts is a manual procedure because there is no device that to do this in an automated way. The procedure is done for each nucleus, measuring the synaptonemal complexes and counting the foci on each synaptonemal complex using a program that gives the distance from each focus to the centromere. For this program gives an Excel sheet per nucleus with the relative lengths and the as percentage of the total synaptonemal complex set and in micrometers, and also the number of MLH1 foci in each bivalent and the position of each focus on the synaptonemal complex arms. This is all in one Excel sheet. In birds, it is possible to count the crossovers or MLH1 foci in one nucleus, the number of foci per bivalent, and to obtain the relative and absolute position of focus on SC arms. But the number of foci and the positions can be obtained with certainty only for the 6 or 8, depends on the species, largest chromosomes, because the rest, they all form synaptonemal complexes that decrease gradually in size as the chromosomes in birds do. So it is possible to find the synaptonemal complex counterpart for each macro SCs only for the 6 or 8 largest chromosomes and the ZW bivalent or ZZ pair. So as most of you know, if you work in science in general, these manual procedures requires a lot of invest a lot of time, and MLH1 counts in birds are not the exceptions. Counting MLH1 foci in birds is especially challenging because of the high number of chromosomes, usually 76 to 80, and the presence of at least one focus in each chromosome, and many times the presence of several foci along macrobivalence. Just to give an example, our former lab students, Lucia, she counted 45,000 MLH1 foci in more than 800 nuclei to analyze the crossover distribution and frequency in 5 species of birds. But once you have this information for a given species or chromosomes, it is possible to do many comparisons, such as, well, you have the total number of crossovers between species, or the distribution of foci along revalence, or to compare the recombination frequencies with linkage maps if they are available for the same species. And because each MLH1 focus represents one crossover, there will be 50% recombinant gametes per focus. So the average number of foci found for species multiplied by 50 gives the size of the genetic map in centimorgans. If a species has on average 60 foci, then the genetic map of the species will be 3,000 centimorgans. And the same can be calculated for individual bivalents or for specific segments of a bivalent, for example, a chromosome arm. Counting the crossovers and scoring the position allow to build these MLH1 maps sorry, that are usually presented in the form of a histogram, like here. This histogram represents the frequency of crossovers obtained by pulling all the MLH1 foci along the synaptonemal complex 1 of the chicken, in this case in 138 nuclei. The intervals of these MLH1 maps can be up to 0.5 micrometers wide, if we take into account the resolution of the measurement on immunofluorescent images. In this particular sample, each interval is equivalent to 3.5 megabases, because the average SC length of the synaptonemal complex 1 of the chicken is 28.5 micrometers, and that gives 57 intervals of 0.5 micrometers. And because Gallus 1 in the genome assembly is 197 megabases, divided by the 57 intervals, that gives about 3.5 megabases per interval. Why is important this information? Because it gives a chance to estimate the crossover rates in centimorgans per chromosome for specific chromosome segments. If we build the cumulative centimorgan map from the telomere of the short arm, and we add all the centimorgans for each one of these intervals, we obtain the total centimorgan map. In this case, 360 centimorgans, or cumulative centimorgan line. So, in the chicken, the MLH1 map crossovers show some characteristics of the recombination in this species. With recombination modules, the presence of more than 3 crossovers is quite frequent in the largest macrobivalence of the species. For example, the average number of foci for the synaptonemal complex 1 is 7, and it is observed that the foci tend to be more or less uniformly distributed along the synaptonemal complex arm. That is, that there is not a critical or accumulation of foci in certain chromosome segments. Another important point is that the average length of the synaptonemal complex that is the x-axis in each one of these histograms. This average length has a linear relationship with the average number of MLH1 foci. That means that the synaptoma complex length predicts the number of crossovers among macrobivalents of the chickens. I would like to point out that chromosome 5, for example, in the chicken, is about 60 megabases, which is the size of chromosome 19 in the mouse. So most chromosomes in birds are smaller than the smallest autosome of a modern mammal like mice. And the mouse bivalent 19 almost invariably has one crossover, but the macro SC5 in the chicken has two, sometimes three. The average is 2 point something. And this simple comparison shows that there is more recombination per megabase in Gallus domesticus macrobivalens than in mouse or human chromosomes. A similar distribution and of the crossovers with several crossover events per macrochromosome is observed in the primitive bird American rhea, and Rhea americana is the ema in Brazil, our nyandu. There's also no obvious localization of chromosomes, of the crossovers on chromosomes, and the SC length also is predicted of the number of crossovers. That means that it is reasonable to think that the presence of multiple crossover events in the macrobivalents in birds is an ancestral characteristic because Rhea americana belongs to the most basal group among living birds, that is the group of Phalignathus birds. Before we studied the MLH1 foci in the chicken and other birds, we observed the recombination nodules in females and male pigeons. As I showed you before, in this bird, the recombination nodules are very clear to observe. And the recombination nodules also has a uniform distribution along the macrobivalence, and the number of recombination nodules is very similar in females and males. So the fact that the combination nodules and MLH1 foci has similar distributions in birds that are distantly related across avian phylogeny gives an idea that these features of the combination in birds are homogeneous and are maintained in spite of the long-time divergence of these species, because, for example, the group of the American rheas and the group of Neoaves is most recent radiation, where we can situate the pigeon, are evolutionary distant as much as 100 million years. And still, the number of crossovers and the average numbers are quite similar and grouped within around 60 crossover per cell. In the middle of these conserved characteristics, we found an exception in the zebrafinch. The zebrafinch is a strinded bird, Passeriformes, belongs to Passeriformes, and has a typical avian karyotype with 40 chromosome pairs and many microchromosomes, but the distribution of crossovers is distinctively different from the other birds that I showed before. In this bird, The MLH1 foci are situated towards the end of the bivalence. Metacentric and submetacentric elements like this one show the foci on opposite ends with large segments of the bivalent without foci, and acrocentric bivalents like chromosome 6, it is here, both in males and females, the same type of distribution, have the foci located on opposite ends of the long arm. This particular crossover localization in the zebrafinch is so extreme that 90% of the foci are observed within 25% of the genome, this at the level of the macro chromosomes. This crossover distribution or landscape, as it sometimes named, leaves large segments of the macrobivalence with very low recombination rates that are represented here by this shaded space in the ideogram. And this creates a significant heterogeneity in recombination rates within the larger chromosomes. This distribution, symmetric distribution of the crossovers, explains the negative correlation between the local rates of recombination and the degree of linkage disequilibrium that was found later in analysis of recombination using single nucleotide polymorphism analysis in this species. In fact, with this particular recombination distribution, we had the chance to look at the consequences of the crossover polarization in the ZZ bivalent, which is this here signaled by the arrow, and with the cytogenetic analysis of the crossover, we use the MLH1 centimorgan maps of this chromosome and the localization of 9 single copy sequences that are linked to the Z chromosome, because localizing the sequences on S spreads gives the chance to count the average number of crossovers in the same chromosome segment, having at the same time the distance in micrometers and in centimorgans. For example, if we take the average distance of the fish signal for SPIN, which is the gene for the spin link that is located on the short arm of the Z chromosome, we obtain a genetic distance using the MLH1 count of about 47 centimorgans. This genetic distance is very similar to the position of the centromere in the genetic map, which is about in the middle of the chromosome. But as I said before, this gene is located near the telomere of the short arm. So the lack of linearity between the physical and genetic distance is a consequence of the accumulation of crossovers near the telomere of the short arm. The consequence is a clustering of markers and linkage markers. And in fact, this is exactly what was observed for the Z chromosome and also for other macrochromosomes in the zebrafinch using linkage mapping molecular analysis, molecular markers, to build linkage map by pedigree analysis. What we see is an accumulation of markers that here surrounded by this line, around 20 centimorgans, when they are probably more or less homogeneously distributed around the Z chromosome. In fact, the linkage map underestimated greatly the genetic distances. The ZZ bivalent has in general 2 MLH1 foci. That means that the genetic length is about 100 centimorgans, but it was only 32 centimorgans in the linkage map, probably due to the difficulties of finding recombinants in the middle region of the chromosome. Something similar happened with the genotyping of single nucleotide polymorphism that give a very high resolution of the recombination in the zebra finch. In these studies, is shown that the shape of the relationship between genetic and physical positions has a long stretch of 0 centimorgans, which is exactly what we observe in the cumulative line here of the MLH1 focus map. Even though the cytological maps of the recombination lack the resolution of the linkage maps using genotyping, the cytogenetic analysis of recombination do not miss crossover events because they are not dependent on marker density as linkage studies are. And this is an example of how the cytological maps of recombination can be used as scaffold or a guidance to linkage mapping when they are available or there is a project to start genotyping in a bird or in another organism. So now I would like to talk about a bit more about the cytogenetic data and what tell us about the global crossover rates in birds and show how they relate to phylogeny. A number of studies have shown that in mammals, there is a phylogenetic signal for recombination rates, with lower recombination rates in basal taxa and higher recombination rates in more recently evolved taxa. So we wanted to know with the available information in birds, we could relate both parameters. This MLH1 foci data can be converted into recombination rates in centimorgans per megabase because we know the DNA content of these birds and we can transform the average number of MLH1 foci into centimorgans. So doing that, we see that the recombination rates are more or less homogeneous when we take global recombination rates, including all the chromosomes. But if only the information for the crossover rates are taken only for macrochromosomes, then we see that, for example, the zebra finch, there is a difference sometimes, more than 50% difference with the recombination in other birds. This is shown perhaps more clearly in this column graph, where the lighter columns are for macrochromosomes 1 to 6, and the rest are global recombination rates for chromosomes. So recombination rates are rather homogeneous, at least for this species that we have analyzed. But the combination rates are especially different when you take only the information for macrochromosomes. The zebra finch was for some time the only bird that had this restricted pattern of with crossover polarization towards the chromosome ends, with jungles of recombination on the end of the chromosomes and deserts of recombination in the middle of the chromosomes. We had the chance recently to analyze the distribution of recombination in the Guinea fowl, which is a species of Galliforme from the Numididae family. that has a similar karyotype to the chicken, the average karyotype for birds, and it's 76. So it has a diploid number of 76, and it has been a long time demonstrated by cytogenetics, Japanese cytogeneticists, that the macrochromosomes of the guinea fowl and the chicken has a few differences, mainly because of a fusion of chromosome 5 and 6 of the chicken that give rise to chromosome number 5 in the guinea fowl. And this sequencing and assembly of the guinea fowl genome in 2019 corroborated these homologies. So in the guinea fowl, we found a very similar distribution of the crossover to the zebrafinch. The localization of the crossovers also towards the ends produces accumulations that gives this U-shape distribution of the crossing over that leaves large segments of the macrochromosomes with very low levels of recombination. In fact, similar to the zebrafinch, 86% of the foci are located within 20% of the length of the macrochromosomes. And the number of oocytes that we examined was similar to the chicken. So this is not a matter of low sampling. Well, in this picture here, you can compare the number of bivalents with a certain amount of foci. In the guinea fowl, it's common that the most macrobivalents have 2 foci, which is represented by this purple color, but only chromosome 6 in the chicken has most frequently two foci. So this shows graphically the difference of the number of crossing overs in the chicken and the guinea fowl. And this is a graph to show the recombination rates that was calculated in centimorgans per megabase in segments that has 4 megabases in the chicken and the guinea fowl. So this segment here have about 4 megabases, and the color blue represents the regions with less than 1 centimorgan per megabase of recombination rates, and which is most of the extension of the length of the guinea fowl, but not in the chicken. So to summarize the studies of emelatron fossa in birds, so far there are 17 species that have maps of the crossovers that were performed using cytogenetic methods, most of them using emelatron, but also Lambert's chromosomes. About 1/2 of these studies were done in our laboratory, and the rest were done by Russian researchers who've done some very interesting works in species, especially of the group of Neoaves. These species belong to 7 orders, but not all the orders are equally represented. If we take the orders with at least 2 species analyzed, it is possible to compare the crossovers within and between different taxonomic levels. So let's do that. And here are these species. I added the American rhea because it's a primitive bird and it's a very valuable information. And it is clear here that the number of crossovers differ more, for example, between the geese and ducks, both species of Galliformes, than between a species of different orders like the Guinea fowl or the zebra finch, which are separated by more than 90 million years of difference time. That gives a first approach that even though the number of crossovers are elevated, people elevated because of the large number of chromosomes in birds, and because most of them have many crossovers in macrochromosomes, there are variations within orders that can even be larger than variations between the orders. Here I choose to represent the similarities and differences using the number of crossovers or MLH1 foci, because these birds have very similar genome sizes. So if we calculate the centimorgan maps and divide them by the genome size, we obtain similar we obtain recombination rates that are also comparable. So we go now to compare the recombination rates in this small sample of birds, a small sample. Well, we confirm that recombination rates are higher, but we can say now that the variation in the recombination rates among birds is much lower than the variations in mammals. If we consider all the mammalian species studies, the recombination rate varies more than 10 times among mammals, but only twice by two in birds. So this is a very narrow variation. Now, the number of crossovers are not related to genome size. That is, larger genome size does not mean that there will be more crossovers. But it is possible that some genomic features, such as lower amount of repetitive elements in birds, have to do with their highest rate of recombination. And for example, the genome size of this species, from the American rhea to the zebra finch, is very similar with very little variation, but the genome size only in primates have more variations than in all these birds that are very phylogenetically different. However, the recombination mechanisms are subject to so many controls at so many levels that there could be a unique feature that explains the difference in recombination rates between birds and mammals. So to summarize, there are certain features that repeat in many species of birds. That is the presence of multiple chromosome events. That may be an ancestral feature because they seem not easy to change because they have been present for over 1 million years. There are higher recombination rates compared to mammals, not only because of the presence of multiple chromosomes, but also for the presence of multiple chromosome events in chromosomes of similar size. Recombination rates do not follow phylogenetic trends. There is not a phylogenetic signal for combination rates among birds, at least with the species that have been analyzed so far. And because of the presence of larger variations across taxa than within taxa. And the karyotype features, for example, fundamental number or the presence of heterochromatin, are not related to crossover variations. So to finish, I'd like to leave some thoughts about the relationship of the stability of the karyotype and the genome, and the recombination in birds. Now, the karyotype of birds has low rates of interchromosomal changes, at least when compared to mammals or non-avian reptiles. So there are a high degree of synchrony even between distantly related avian lineages. This creates an apparent paradox between the karyotype and the genome conservation and the morphological and adaptive diversity of the many species of birds that are found populating very different environments. It has been suggested an explanation possibility that make compatible these two facts, and is that the organization of the bird genome in numerous chromosomes favors the genetic diversity, increasing the capacity for random segregation due to the presence of multiple linkage groups or chromosomes. The MLH1 data supports the idea that the karyotype homogeneity does not restrict the advent of new combination variants, such as localized versus non-localized recombination. So, in other words, the variant recombination landscape that are observed on macrochromosomes, restricted versus non-restricted recombination, also creates genetic variations, which contributes to the great phenotypic diversity that is observed in almost 10,000 species of birds. So I would like to thank again to the organizers, and I'll be happy to take any questions.
Muchas gracias otra vez, María Inés Pigozzi, por tu fantástica presentación. Ahora yo voy a pasar la palabra para mis colegas, mis compañeros, para que conduzcan las preguntas.
Bueno, muchas gracias, Inés. La verdad que increíble, está buenísimo que alguien se ocupe de la meiosis, porque nosotros los que hacemos más mitosis es el lugar donde ocurre todo y como que pasa desapercibido, y es lo más importante que hay que estudiar, ¿no es cierto? Se estudia cualquier cosa de citogenética. Yo tengo una pregunta más que nada haciendo referencia a la variación intraespecífica de la recombinación dentro de un mismo sexo. Hay estudios que lo ven eso en sentido, ven la variación intraespecífica que hay dentro de la recombinación, apuntando a que existen factores que inducen la recombinación, o por lo menos lo leí por ahí, como los cromosomas B, que en presencia de cromosomas B, por ejemplo, aumenta la recombinación. ¿Se tiene en cuenta eso cuando se hacen así análisis filogenéticos, la variación dentro de la misma especie?
Sí, sí, por supuesto. Es más, hay algunas especies en las que se ha observado dimorfismo de la recombinación según el sexo. En los propios, o ya sea especies domésticas o especies, digamos, silvestres. También es interesante, al menos hay un estudio que compara la diversidad de la recombinación, niveles de recombinación en machos y hembras en dos especies, unas con más dimorfismo y otra con menos dimorfismo sexual. El análisis comparativo, hay pocas especies pero analizadas macho y hembra, con características, bueno, dimórficas o sin dimorfismo sexual, y no hay una relación en las aves. No parece haber una presión selectiva hacia favorecer el aumento o la disminución de la recombinación según el dimorfismo sexual, pero sí hay algunos estudios, y al menos un estudio de la variación de la recombinación en distintas líneas comerciales de pollo. Pero ya son estudios, digamos, más chicos, porque, bueno, se necesitan muchos animales realmente para,
claro, no, me imagino.
comparar, ¿no?
Sí, más que nada, Sí, mi pregunta es más que nada, porque desde ignorante pregunto, ¿no se sabe bien cómo se induciría la recombinación cuando hay algún, o sea, no se sabe bien cómo actúan esos inductores que algo se
Claro,
dice?
no, bueno, sí, bueno, en cuanto a la distribución, digamos, de la recombinación que favorece, hay, en aves hay un solo estudio que, es un estudio en dos, finches, uno de ellos, el zebra finch, que muestra que los eventos de crossing over aparecerían con más probabilidad ligado a secuencias funcionales, sea cerca de reguladores de genes o cerca de genes. Eh, Se ha visto, al menos en ese único estudio que hay, que los sitios donde más probablemente habrá recombinación son los hotspots. En aves son más estables precisamente porque están asociados a secuencias funcionales, algo que es muy distinto a lo que pasa en los mamíferos, donde las secuencias están señalizadas por bueno, proteínas que favorecen la unión de PRMD9, que no necesariamente se une a regiones que tengan algún significado funcional. Por eso en los mamíferos, los hotspots o sitios donde más probablemente se produce la recombinación son muy variables, cambian entre una especie y la otra, son muy dinámicos y en las aves serían más estables. Eso, digamos, pero ese es a nivel de alta resolución de la recombinación. Yo he estado ahora con algunas mm, informaciones que hay más genómicas, tratando de ver si la concentración de crossing over, por ejemplo, en zebra finch o en la gallina de Guinea, se relaciona con la presencia de densidad de genes o secuencias reguladoras. La verdad es que no es así, o sea que digamos que la relación desde los hotspots, que son a nivel de kilobases, hasta lo que uno ve como hot region de la recombinación, que es a nivel de megabases, no es fácil de explicar.
Sí.
No es sencillo, pero sí hay algunos estudios, no a nivel citogenético, sino más bien a nivel de alta resolución de recombinantes o de polimorfismos a nivel molecular.
Gracias.
María Inés, bueno, muchas gracias por la palestra, siempre excelente. Tu trabajo de tantos tiempos, desde que saliste de Misiones, trabajando con la meiosis, es realmente un placer poder escucharte. Cuando yo siempre pienso en aves, por los años que también tengo trabajando en aves, las aves nos plantean una dificultad, un desafío enorme para primero poder tener muestras y después poder plantear en genética el tipo de estudio que uno haga. Entonces, cuando uno piensa en mitosis hay una serie de dificultades y cuando yo pienso en meiosis, los desafíos son mucho mayores. Vos nos has mostrado una serie de datos excelentes con algunas o básicamente especies con las cuales se tiene un acceso más fácil, son domésticas o peridomésticas, en fin. En ese sentido, la pregunta es, ¿existe algún grupo de aves o algún grupo de especies que te plantee algún desafío interesante que quieras o que deseas estudiarlas y que aún no has podido o que sea interesante?
Sí, seguramente las aves que tienen cariotipos más modificados serían interesantes de a ver cómo afecta los que tienen cariotipos, más rearreglos intercromosómicos, porque la baja tasa de arreglos cromosómicos, digamos, en las especies por lo menos que están estudiadas, la mayoría, eso podría oscurecer, digamos, algún rol del cariotipo en variaciones de la recombinación. No es así porque la gallina de Guinea, digamos, es un ejemplo que resultó ser muy interesante porque dentro de un grupo extremadamente homogéneo de inmunidades, facianidades, porque los crácidos, por ejemplo, serían muy interesantes de ver para seguir completando un poco el panorama de Galoancerae. Pero sí las especies con menor cantidad de microcromosomas, hay una especie estudiada que es Falco subbuteo, que el grupo de Rusia, ellos han estudiado varias especies silvestres porque tienen pocas restricciones. Si las aves no están, digamos, catalogadas como en peligro en determinados niveles, lógicamente como hay que sacrificar las aves para hacer los estudios meióticos, bueno, realmente conseguir esos permisos, por lo menos en Argentina es muy problemático, pero ellos trabajaron con un grupo también cerca de Novosibirsk, que es un grupo que, de recuperación de fauna silvestre. Entonces, algunas de las especies, sobre todo de falco, y las estudiaron porque eran animales injured, ¿no? Que tenían algunas lastimados y que o hubo que sacrificarlos o murieron y estaban trabajando en colaboración con ese tipo de organizaciones, que esa sería, digamos, la manera de acceder, porque es cierto que se pueden hacer estudios de la distribución de la recombinación mediante desequilibrio de ligamentos. Yo no soy una experta en eso, no lo conozco, pero realmente requiere estudios poblacionales muy grandes durante mucho tiempo. Es un esfuerzo enorme porque también hay que seguir las aves a campo, realizar realmente las genealogías, bueno, mostrar los nidos como corresponde, establecer los parentales bien, si bien es posible hacerlo. Y también está el tema de que ese tipo de aproximación a veces muestra dónde hay más variación en el genoma. Entonces uno no termina estudiando todo el genoma. El mapa de ligamiento, otra aproximación, ¿no? Pero el mapa de ligamiento del zebra finch se publicó el mismo año que nosotros hicimos ese pequeño análisis de distancias genéticas y físicas en el cromosoma Z, pero quedó así, incompleto, con 30 centimorgans cada Z, con menos crossing-overs que cromosomas. Yo estuve con uno de los autores y dice, sí, nosotros la verdad que tendríamos que haber puesto al menos 50 centimorgans porque sabemos que hay un crossover obligatorio. No, o sea, eso yo lo hago la equivalencia de que si uno va y publica, no sé, un año cariotipo, por ejemplo, de ñandú y dice, bueno, tiene 70. Y al otro año dice, encontré 4 más. Tiene 74. Creo que a nosotros nunca nos aceptarían en ningún momento de la historia de la citogenética, nos hubieran aceptado llegar al número diploide correcto a plazos. Bueno, como los estudios de ligamientos son muy dificultosos, yo entiendo eso, realmente ir avanzando sobre segmentos del genoma o de los cromosomas, eso es algo aceptado, pero muchas veces crea así percepciones sobre, por ejemplo, las tasas de recombinación que no son correctas y que después muchas veces no se corrigen.
Correcto.
Siguiendo acá, Daniel bueno, te felicita por la excelente charla. Y bueno, haciendo referencia a la distribución conservada, relativamente conservada de los sitios de recombinación en los macrocromosomas, pregunta si existe alguna evidencia relacionada con la presencia de hotspots de recombinación asociada con regiones ricas de ADN repetitivo, como por ejemplo relacionada a las NORS. Y después dice, ¿consiguieron identificar alguna evidencia de la distribución de los nódulos de recombinación en estas regiones?
No, la respuesta es no. Con respecto a determinadas secuencias, no, que era algo a lo que me referí así antes, que digamos, hacer el link entre un determinado tipo de secuencia y una determinada frecuencia de recombinación, eso no lo hemos hecho. Solamente sí un pequeño ensayo que publicamos ahora sobre la presencia de, bueno, recombinación más alta en una región subtelomérica en el pollo. Eso ocurre frecuentemente, que hay una mayor frecuencia de nódulos de recombinación, focos de MLH1, con contenido GC más alto. Eso es algo que se conoce, la relación entre regiones ricas en GC y mayor tasa de recombinación. Eso es un trabajo que en realidad lo hicimos con otro objetivo, digamos, que era mostrar cómo el complejo sinaptonémico, digamos, está sobre representado, digamos, en las regiones de mayor recombinación, tal vez porque hay lazos de cromatina más cortos en esas regiones que favorecen la aparición de, bueno, o la resolución de más cantidad de crossing over. Pero no lo hemos hecho y tampoco no hay hecho en aves. Ahora lo que se está haciendo, así como para mostrar ese tipo de asociaciones, son el análisis de Topological Associated Domains, de TADS. El análisis de TADS en células meióticas, en organismos donde se conocen los hotspots, se mapean las regiones que pueden tener más asociación en un determinado momento cuando se inician las interacciones de recombinación y se estudian, digamos, cuál es la composición de esas regiones. Eso se ha hecho, sí, en el ratón.
Yo tengo una pregunta, puede ser ahí. Te felicito otra vez por la charla y te pregunto si hay algún estudio que muestra la conservación de los fósiles de recombinación cuando tenemos un cromosoma que es conservado entre especies. Es decir, si el cromosoma 1 de gallina hay 5 sitios de recombinación y está en otro pájaro este mismo cromosoma y también hay 5 sitios, 5 puntos de recombinación.
Bueno, eso justamente es lo que trató de mostrar, pero recién ahora estamos yendo un poquito más a relacionar micrómetros y megabases con lo que pasa en la gallina de Guinea y el pollo, Gigalus domesticus. Son realmente los macrocromosomas tienen un grado muy alto de sintonía, incluso en la misma dirección, y las realidades que la recombinación varía muchísimo. No tenemos, por lo menos a nivel de genoma, realmente, si uno los pone en paralelo, no hay a nivel de secuencia algo que explique esa variación. Después, seguramente, como hay tantos elementos que intervienen en la regulación de la recombinación, la epigenómica en las gametas, eso es algo que no está estudiado en ningún ave y no está del alcance, digamos, por lo menos en el plan de hacerlo, porque está muy lejos del tipo de estudios que hay que hacer, porque creo que lo más correcto sería encontrar sitios de hotspots en una y en otra especie, y eso es aún nivel que no es de cromosomas, sino que es de genoma a nivel de kilobases, digamos, que hay que encontrar. Pero si se imaginan que encontrar los hotspots en dos especies de aves, ese es el trabajo que se ha hecho, que salió publicado en Science y demás. Realmente no es un trabajo sencillo de hacer, ni que haya mucha gente tampoco que tampoco no hay tanta gente obviamente que lo quiere hacer. La mayor parte de los estudios están enfocados en organismos modelo. El ser humano no es un ave, somos mamíferos, entonces muchos de estos estudios, seguramente por cuestiones de financiación y demás, se hacen en, en modelos de mamíferos. Y también que a veces en las aves hay estos estudios están más ligados a la parte ecológica, adaptativa, de dimorfismo sexual, y por eso se estudian por ahí los específicos o en algunas especies de aves, pero no, no hay hasta el momento una forma de ligarlo fácilmente.
Sí.
Aprovecho
No.
para hacer otra pregunta muy corta. Si es posible mirar una correlación directa, por ejemplo, entre los cromosomas sexuales, por ejemplo, los paleognatas que tienen cromosomas sexuales casi iguales, no hay casi ninguna diferenciación, y las otras aves que son más modernas, así decir, donde tenemos los cromosomas sexuales mucho más diferenciados, se mira un estrato de diferenciación de la presencia de los quiasmas que están se desplazando al final del cromosoma, si no hay ninguna relación con acúmulo de ADN repetitivo en los cromosomas sexuales, su degeneración y su condición para hacer el crossover.
Bueno, en las paleognatas, nosotros estudiamos dos especies. La distribución del crossing over en el segmento pseudoautosómico, ¿no? En el segmento recombinante del ZW de las dosis de REA americana y de REA pennata, son iguales en densidad, en distancias entre eventos de recombinación, promedio de distancia entre eventos de recombinación, que autosomas de similares características. Es más, hay un trabajo que está hecho en el EMU y que muestra que no hay tampoco o sea, por megabase menos recombinación, o sea, como que no hay elementos disuasorios de la recombinación, elementos que podrían estar disminuyendo la cantidad de recombinación en esas regiones recombinantes grandes. O sea, la distribución, tenemos hecho, pero no está publicado, un pequeño estudio de distancias entre crossing over, sería de interferencia de crossing over, y no hay diferencias significativas de interferencia de crossing over en el segmento pseudoautosómico, en el ZW de REA, que en un autosoma de la misma especie, digamos. Después, claro, sí hay en otro plano que no tienen que ver con estudios de la recombinación, sino con estudios de la diferenciación genómica, ¿no? De la diferenciación de secuencias en esos cromosomas, que sí están muy bien estudiados, digamos, pero el crossing over tampoco no está disminuido, digamos, más hacia un determinado sitio dentro de los sexuales más homomórficos, como el de las reas.
Sí, así como mi conclusión por lo menos parecería que, bueno, la gallina de Nueva Guinea y el zebra finch rompen toda la estructura, el patrón filogenético que se podría llegar a sacar dentro de lo más o menos conservado. Así que no, nada más que felicitarte, la verdad que muy interesante lo que haces y sirve muchísimo, la verdad que está buenísimo.
Bueno, gracias, Martín.
Muchas gracias otra vez entonces, María Inés Pigozzi, por tu charla. Estoy muy encantado por estar 1 hora y media más o menos hablando de meiosis, recombinación y pájaros. Estamos muy contentos.
Les agradezco mucho a ustedes, los felicito por esta iniciativa de hacer la reunión de esta manera y bueno, vamos a seguir en las siguientes palestras para bueno, seguir aprendiendo todos. Muchas gracias.
Muchísimas gracias.
Gracias, María Inés.
Bom, pessoal, obrigado então pela presença de todos. Informo para vocês que o nosso próximo webinar vai ser dia 9 de junho, às 14 horas também, né? Já temos um horário meio fixo, né, de nossos encontros aqui. E o título da próxima apresentação será Mapeamento óptico para investigação e caracterização de rearranjos estruturais no genoma. Teremos a doutora Juliana Fonte Mazeu de Araújo, da Universidade de Brasília, e o doutor Christopher Michael Grokowski, que é do Molecular and Human Genetics Baylor College of Medicine dos Estados Unidos. Obrigado a todos, uma ótima tarde e um resto de semana. Tchau, tchau.